Hello, and welcome back to Introduction to Genetics and Evolution. In the previous videos, we've talked about deviations from the Hardy-Weinberg equilibrium. The most recent videos, we talked about natural selection. This is very predictable changes in response to differences in fitness. Now, one of the assumptions of Hardy-Weinberg that I mentioned before was an infinite or near infinite population size. Obviously, there are no species out there that have an infinite number of individuals. As a result, there will be some sampling error, or as this is referred to here, genetic drift. In this video, we'll talk about the effects of sampling error over single generation. Now, contrasting natural selection with genetic drift, natural selection is highly predictable. You have a subset of genotypes that have higher fitness than other genotypes. This higher fitness is associated with having more offspring, and as a result, these genotypes become overrepresented. Now, if you know the fitnesses associated with the different genotypes, then the changes in allele frequency associated with natural selection are very highly predictable. But not all evolutionary change is this predictable. As I mentioned, species typically have finite numbers of individuals. As a result of that, random chance matters. Let me start with an analogy. Let's look at a bag of marbles. Let's imagine that you have brown marbles and blue marbles only in this bag, and exactly half the marbles in the bag are brown, exactly half the marbles in the bag are blue. Now what's going to happen is you're going to start a new bag of marbles. Now what if we start this new bag of marbles with exactly 4 marbles from the old bag? And we just reach in and randomly pick out 4. How many of each color would you get? Well, you could get 4 blue, you could get 4 brown, you could get 2 blue and 2 brown. Now, probabilistically, there's only about a 5% chance that you get all 4 marbles the same color. You'd either get 4 blue marbles or 4 brown marbles. Not very likely. So you're going to have a mix of colors in the new bag that you start. So this is associated with having 4 marbles starting the new bag. Now what if you started the bag with even fewer? What if we started with exactly 2 marbles? Well, how many of each color would we get? Well, there's 3 possibilities. 2 blue, 2 brown, or 1 blue and 1 brown. And in fact, in this case, there's about a 50% chance that the 2 marbles we'd get would be the same color, that we'd either get 2 blue marbles or 2 brown marbles. Well, this illustrates the point of sampling error. So this prior example, when you picked 4 marbles, it's likely you'd get roughly the same proportions as you had in the previous bag, right? So this would be the right proportions, that it mimics the previous generation. If you pick 2, you're very unlikely to get even roughly the right proportions. So by picking more, you tend to get a more representative sample of the original pool. This is the principle that we see in general. If you want to understand if people in a particular supermarket are tall, you don't just look for one person, but you look for a lot of people. So you get a representative sample. Well, the same principle applies in nature. Again, populations are not infinite, and frequently small samples are not perfectly representative, and these are the ones that form the next generation. Because they're not perfectly representative of the previous generation, the allele and genotype frequencies change between these generations. And this effect can compound more and more over time. We'll talk about the compounding effect over many generations later, but let's focus instead on what's happening in one generation. So it's random in direction over one generation, whether you're going to increase in frequency or decrease in frequency. So assuming that there is more than one allele, so assuming you have at least 2 alleles, any allele is about equally likely to increase or decrease in frequency after one generation of sampling error or genetic drift. So for example, if the allele frequency of big A, assuming there's 2 alleles, big A and little a, if the allele frequency of big A is 0.6, you're about equally likely to have frequency above 0.6 or below 0.6 in the next generation. However, it's very unlikely you're going to have exactly 0.6 again, because there is a decent chance you're going to have some non-representation. Even if you're having a very large sample, it might end up being 0.603 or something like that. So allele frequencies tend to drift due to this sampling error, and this is where the term genetic drift came from. Let me show you what would happen in the context of this. Imagine tossing a coin 10 times. So this is similar to having p equals 0.5. You may get 5 heads from these tosses, right? So that would be exactly what's expected in the sense of the average, right? Since you're equally likely to get heads or tails. Now getting more than 5 heads or getting fewer than 5 heads is equally likely. This shows you the distribution that you expect, the probabilities. Probability of getting exactly 5 heads is actually less than a quarter. Your probability of having 10 heads is extremely low. Probability of having 0 heads is extremely low, but you're about equally likely to have slightly more or slightly less than 5 heads when you toss the coin. Okay? In fact, actually, the probability of getting 10 heads is about 1 in 1,000. It's very unlikely, unless it's a weighted coin. Well, the same concept applies to populations. So if your original population has an allele frequency of big A of 0.6, you see below what would happen in one generation of genetic drift. The probability of having it greater than 0.6 versus less than 0.6 is about the same. And this is the case if you have 10 diploid offspring. Okay? Now, let's see what happens as we look at this over multiple generations briefly by just looking at the individual steps. So this magnitude of change compounds as it relates to population size. Now, greater changes are going to occur if the population size is smaller. You'll have greater individual deviations in allele frequency per generation as the population is smaller. So let's look at 3 different population sizes. Let's look at population size 400. You're starting, in this case, this is looking at generations on the x-axis and allele frequency on the y-axis. You're starting at 0.5. And what's happened here is we're starting 1, 2, 3, 4, 5, 6, 7, about 8 different populations at 0.5. And this shows random changes over all 8 populations. After about 100 generations, we see there's a few that are kind of close to 0.5. Some of them are higher, some of them are lower. So we have this genetic drift that's compounded over time. This shows, this little green figure shows the approximate average size of a change in one generation in any of these populations. Now let's see what happens if we reduce this. Instead of 400, what if we looked at a population of size 40? What we see in this case, much bigger changes. We have some alleles that have gone all the way to 100% or 0% in some of these populations, and some are still segregating. But again, you're equally likely to go up or down in any individual generation, and the individual step size now is much bigger. You get bigger random changes in allele frequency with smaller population sizes. Right? Let's do the same thing. This was with population size of 40. Let's look at it in the extreme with population size 4. Wow, all variation is lost. We'll come back to this actually very shortly. But you see, the individual step sizes here is very large. Again, in each of these cases, these are 8 different simulations, each with population size 4 and starting the allele frequency at 0.5.
So with those examples in mind, can we solve mathematically how big the individual steps are that result from genetic drift in a single generation, on average? The answer is yes. So we can use the variance. Now recall, we used the variance before when we were studying heritability a couple of lectures back. We look at the variance in allele frequency due to one generation of genetic drift. So looking at how much of a spread is there. The answer to that is PQ divided by 2N, where P and Q are the 2 allele frequencies, and N is the population size. We use 2N because we tend to work with diploid organisms. Now, how do we use this to actually look at average changes? Well, we can look at the standard deviation. The standard deviation of this would be an estimate of the average allele frequency change in one generation. And in fact, mathematically, it actually would be a slight overestimate, but it still gives us an idea for illustration purposes. So how do we get the standard deviation from the variance? Well, the standard deviation is always the square root of the variance. So we have this formula right here for the variance. We take the square root of that, as illustrated here, and that gives us the average allele frequency change from one generation of genetic drift. So let's apply this to the examples that I just showed you the figures from a couple of slides back. When we had a population size of 4, we had starting allele frequencies, 0.5 and 0.5. The average change based on this formula should be about 0.18. What this means by the average changes is if you start with an allele frequency of 0.5, it's likely that you will go up to about 0.68, or likely that you might go down to 0.32. That's sort of an average change. The change could be more than that. You could go to 0.70, you could go to 0.62, so maybe it's larger, maybe smaller of a change. It also could be up or it could be down. We don't know the direction of genetic drift from one generation. But this gives you an idea of the average step size. Okay? Now notice for this one is 0.18. If a population size is 40, the average change is quite a bit smaller, as we witnessed. In this case, the average change would be only an allele frequency change of about 0.06. If the population size is 400, the average change in one generation of genetic drift is 0.02. Now, you notice with those very small ones, like the population size of 400, That's why in the example, when we're looking at a population size of 400, no allele was ever lost or fixed. We always still had variation in the population because the individual steps were very small, and even over 100 generations, we still retained variation in that population. In contrast, when we looked at the very small population size of 4, we lost all variation very quickly because the step sizes were very big, and it was very likely to get to 100% or 0%. Okay? So what are the take-home messages from this lecture as a whole?
So here are the take-home messages from this video. Drift is strongest in small populations. Drift is neither predictable in direction nor exactly replicable in degree in one generation. That you saw with all those different populations, even when we started it over and did it again, they didn't all follow the exact same track. They all had the same, on average, change in allele frequency, but since some went up, some went down, some had a little bit more than average, some had a little bit less than average at times, it's not exactly replicable in degree, and it's not predictable in direction in one generation. Very important addendum there. And finally, drift can cause big changes in allele frequency over time. We'll pick up on this in the next video. Thank you. Hello, and welcome back to Introduction to Genetics and Evolution. In the previous video, we talked about the effects of genetic drift over a single generation. I emphasized the point that in small populations, genetic drift can have large changes in allele frequency. I also emphasized that in a single generation, you cannot predict whether genetic drift is more likely to make the allele frequency go up or go down. What we'll do in this video is talk about what happens with genetic drift over many generations, and also look at the interaction between genetic drift and natural selection just a bit. Now again, in terms of this long-term effects of genetic drift, you saw this graph in the last video. If we start with a variable population that has 2 alleles, the x-axis here is generations, so this is going out 100 generations. The y-axis is allele frequency. We're starting 8 different populations with an allele frequency of 0.5. So big A has a frequency of 0.5, little a has a frequency of 0.5. What you notice is that after many, many generations, if we extended this out, not 100 generations, but thousands of generations, the allele frequency of A, big A, in each population will either be 0 or 1. So we're not going to have any variation left. That basically what's going to happen is basically the whole population will be big A, or the whole population will be little a for every one of these populations. The reason is that once you get to an allele frequency of 0 or 1, there's no variation. One allele is just gone. If the allele is gone, you can't drift back down. You need a new mutation to come in to reintroduce it. But just working with the variation that's present, genetic drift cannot have any further change. So the long-term effects of genetic drift are to remove variation from a population. Now let me ask you this question. I said in one generation we cannot predict whether an allele frequency will go up or go down. Well, let's imagine we start a variable population, all right? So it has big A's and little a's. There's 2 alleles there. Now let's say the starting allele frequency of big A is 0.6, of little a is 0.4. Okay? What do we expect after a very long period of time? If we were to wait thousands of generations, do we think the population is more likely to be fixed for big A or more likely to be fixed for little a? Which one will go to 100%? Will everybody be big A, or will everybody be little a? Well, that was a little bit of a trick question for you. Now, I mentioned that in one generation, we cannot predict whether you're more likely to go towards more big A or less big A. But what's happening here is we're looking at the effects over long times. Now, there's an analogy we can draw to this. Imagine a blindfolded person walking aimlessly around. If you start right in the middle, and let's say, for example, this person can only go left or right, and they just take randomly steps this way or steps that way, sometimes bigger steps, sometimes littler steps. If you start right in the middle, you're about equally likely to run into this as you are to run into this, right? Okay, well, let's change this a little. Instead of having this person start dead center, as we start the person over here, where they're about 3/4 of the way over to one side. Are they more likely to eventually run into this, or more likely to eventually run into this? Even if they're stepping at random, since this is closer, they're actually eventually more likely to run into this than they are to run all the way out here into that, if they're just moving around aimlessly. Not convinced? Well, how about this one? Let's say you start right there, and they're taking random steps in 2 different directions. Well, they're pretty likely to run into this end over here, whereas they're very unlikely to run into this end way over there. So this is a funny thing about genetic drift. Now, the probability of a long-term outcome is actually somewhat predictable. So even though in one generation there's a roughly equally likely for an allele to go up or down in frequency, that the frequency of big A may go up, may go down, the long-term loss or fixation of allele is actually more predictable. That again, if you start dead center, you're about equally likely to hit either of the two ends. Think of that blindfolded person. If the frequency is much less than 0.5, then it's likely for the allele to be lost. You're likely to run into that zero end. If it's much greater than 0.5, you're more likely for that allele to be fixed. Well, there's a very simple relationship you might have already anticipated. The probability of eventual fixation, and again, this is over the long term, not one generation. Probability of eventual fixation of allele big A will equal the allele frequency of big A. So if your allele frequency is 0.9, your probability of hitting that 1.0 end is 90%. Your probability of hitting the 0.0 end is 10%, or 1 minus your allele frequency. So let's look at this. Here's 4 sample runs of that program I showed you before, allele A1. Our starting allele frequency is 0.75. So you're towards this end of the overall cage. Again, 3 out of 4 times it's bebopping around there. 3 out of 4 times it ends up hitting 100%. 1 out of 4 times it actually bebops far enough down that it hits 0%. I actually didn't manipulate the data. I actually just ran this as the same do 4 sample runs, and this is actually what it came out with. So as you can see, this probability of eventual fixation is equal to the allele frequency. Now notice there is still some chance the allele will be lost, even if it starts off very abundant. And you see this happen in this case. Even though it started at 75%, it bebopped around, and after a few sudden losses, it did actually go to 0. So this does happen, but this frequency tells you the probability of it. Now let's turn this around a little bit. Let's think of it in the context of species versus population. Everything we've looked at so far has been thinking of what's happening in one population and focusing just on that. Now what happens if we're looking at a whole species that has many isolated populations? So let's imagine we're looking at, for example, the Galapagos land snail, which is found on various of the Galapagos islands. Well, here's a thought question for you. If there are 4 populations of Galapagos land snails, and each individual population is started with an allele frequency of 0.75, what would the allele frequencies be in the populations many years later? Well, you probably can guess that some are going to fix, some are going to be lost, and in fact 75% of them will fix, right? 25% will be lost. So if it's 4 populations, we expect on average 3 to be fixed, 1 to be lost. The second one is something I want you to actually answer here in an in-video question. What would the average allele frequency across all populations be many years later? Let's imagine the 4 populations had approximately the average size. They eventually do their thing. Some are fixed, some are lost. In the species as a whole, so averaging what's happened in each of the 4 isolated populations, there's no migration between the populations or from anywhere else. What will the average allele frequency of big A be? Well, I hope that wasn't too difficult. So, well, again, this is the pattern that we expect to have happened. So some of these allele frequencies will be bopped around. So let's say, for example, here are the 4 islands: Island 1, Island 2, Island 3, and Island 4. Okay. We see in 3 out of the 4 islands, Islands 1, 2, and 4, the allele big A is fixed, which means everybody on that island is big A, big A. And 1 island, little a is fixed. So everybody on this island is little a, little a. So these ones are all big A, big A. That one's all little a, little a. What is the allele frequency across the species as a whole? Well, in this case, we're saying that all of these islands have the same average population size. What we have is 1 island of all big A, big A's, 1 of all big A, big A's, 1 of all big A, big A's, 1 of all little a, little A's. So the species as a whole, even though each individual population has no variation, the species as a whole still has some variation, right? Because the species has some islands that are all big A, and some islands that are little a, little a. And in fact, in the species as a whole, the allele frequency is still 0.75. So it actually hasn't changed in the species as a whole when you've isolated off these islands. So some points to remember is that drift eventually leads to allele fixation or loss in every individual population. Very important facet to it. Now again, the starting allele frequency, or P of big A, is the long-term probability of that allele's fixation. Conversely, 1 minus that should be the long-term probability that allele is lost. But if you have a species with many isolated populations, even though you're going to have some populations fixed for big A, some fixed for little a, the overall species still retains variation with the same P of big A, with that same starting allele frequency. That's a very interesting low facet. Now I want to go into something a little bit related here. What happens when we look at the interaction between natural selection and genetic drift? And specifically, can genetic drift make alleles that are bad spread or even become fixed? Okay, so this is the interaction between drift and selection. Well, this is going to depend on the population size. If the population size is very small, which in that case, again, when the population size is small, those are the times when drift is strong, then genetic drift can sometimes counteract weak selection to make bad alleles spread and potentially even go to 100%. Now, it won't always do it, because remember, drift is a random force, right? That'll sometimes make alleles go up in frequency, sometimes make them go down. And so drift is actually random, but it may cause bad alleles to fix, but it may also push in the same direction as selection. So imagine selection is this constant force pushing down. Let's say it's a bad mutation. Selection is a constant force pushing the allele frequency down. Drift, if it's strong, will be a random force, sometimes pushing it up, sometimes pushing it down. Well, let's look at what happens when you have strong drift and weak selection. What I've modeled here are 6 simulations. This is using allele A1. What I did is I said that the fitness of the big A genotype is 0.98, whereas the fitness of big A and little a is 1.0. So selection is trying to eliminate the big A allele, and it's doing so kind of slowly. Population size here is 100, so it's not a very big population. This is kind of a small population. This would be a case where selection is kind of weak. You know, there's only a 2% reduction in fitness to just the homozygote, so this is a recessive detrimental. There's a 2% reduction in fitness to just the homozygote, but we have very strong genetic drift. So our starting allele frequency is 0.5. What I did is I started 6 populations this way. We see this first one, you know, it bebops around, but eventually big A is lost, as you expect from selection. Here it bebops a lot more, eventually big A is lost. Bebops some, big A is lost, bebops some, big A is lost. It was a funny one. 1 out of 6 of these actually had a bebop up to the point that everybody's big A. So you've actually lost the better allele. Little a is the better allele. But in this case, you fixed the big A type. That's kind of funny. And here we've lost big A. So this is a case where genetic drift actually overpowered selection. In some cases here, this one right here probably is a case where genetic drift actually made selection faster. Because you notice that there's a fairly rapid drop. Here, genetic drift slowed it down, and here, genetic drift actually went in opposition. So drift just kind of comes in and monkeys with what selection is trying to do. Now, what would happen, this is a case of fairly strong drift, what would happen if the population size wasn't 100, but was 1,000, meaning there's a lot less genetic drift? In this case, we see a fairly consistent pattern that pretty consistently the allele frequency of big A is going down, going down, going down, going down, going down, going down. We see a little bit more noise in the system than if it was from selection alone, but there's a very consistent trajectory, and we assume if we extrapolate out further, in all 6 populations, the allele will be lost. This is a case where the drift is weak and selection is also fairly weak, but drift is much weaker than selection, that it's not monkeying what selection is trying to do. Now, there are some extreme cases of genetic drift. One example of these is what's referred to as founder effects. This is a strong example of genetic drift when a new population is established by a very small number of individuals from a larger population. They're often non-representative of that original population. You see this often in the case of colonizing islands, and this can cause the spread or even fixation of detrimental alleles. So imagine you have this population here with red squares and blue circles. And if we randomly take 5 individuals, every now and then we'll get an all-blue population or an all-red population. These are non-representative of the original population, whereas this one right here is fairly representative. Now, this has been observed several times in humans. Here are some examples of diseases or traits that are common in certain populations because of past founder effects. So polydactyly, or having that 6th digit, is common in the Amish. Tay-Sachs disease is actually very common in Ashkenazi Jews. So people like Leonard Bernstein, the famous American composer, Albert Einstein, and Sigmund Freud are some examples of Ashkenazi Jews. Interestingly, the Ashkenazi have maternal ancestry traces back to just 4 women. Right? So this is for about half the Ashkenazi out there. They have their maternal ancestry tracing back to just 4 women. So this was a very strong founder effect associated with them. Huntington's disease is interestingly very abundant in Mauritius. It's thought that one of the original founders was either had the disease or was heterozygous for it. And red hair is supposed to be common among the Irish as a result of it. So just to wrap up, some points to remember: in small populations, Genetic drift can sometimes overpower weak selection. So remember, genetic drift is random in direction. So if you have strong drift and weak selection, you can actually spread or fix bad alleles because of genetic drift. And founder effects are this interesting special case of genetic drift. I hope that was interesting. Thank you for joining us.